It's a real pleasure to welcome all of you to our first Research Exchange live event, and an honor to be introducing our presenters, Bibi Bielekova and Peter Kosa from the neuroimmunology section at NIH, and to introduce our moderator, Mike Rackey. Mike's the medical director for neurology at Quest Diagnostics. He actually started his research career at the neuroimmunology section of NIH, has been a pioneer in MS research at Washington University, University of Texas, and finally at the Ohio State University, where he was chair of neurology. Mike's actually the reason that we are doing our first research exchange, that it was built for the MS community. We launched almost 3 years ago with the simple idea that we needed a new way to connect expert communities to discuss emerging research and to capture that conversation. And when we took our first prototype to Mike about a year ago, he immediately got our vision. And one of the first things he said to us is, "You got to meet June Halper." And like Mike, June immediately understood what we were doing and graciously invited us to participate in the CMSC conference last spring. And that led to our collaboration with Banner Life Sciences, the sponsor of the MS Research Exchange and today's event. Throughout this time, Mike has been all in, both as a member of our scientific advisory board and as the editor for the MS Research Exchange. This event really embodies exactly what we envisioned when we began, which is connecting communities of experts and fostering conversation around emerging research. So without further ado, let's get the conversation started. I'm going to turn it over to Mike. Thank you.
So thanks, Bob, and it's my pleasure to introduce today's presenters. Peter Kosa is a staff scientist in the neuroimmunologic disease section, and Bibi Bielekova is the chief of this section. And I should make one correction. Bibi and I know each other for over 25 years. We both did our fellowship in the neuroimmunology branch, which was a part of NINDS. And now Bibi is a section chief for neuroimmunologic diseases in NIAID. But I should note that over 20 years ago, Bibi and Roland Martin organized a meeting on MS biomarkers in Washington, D.C. And since that time, she really has become a leader in this field. And the paper that Peter and Bibi are going to discuss today, I don't want to say is a culmination of that work, but I think gives you an idea, will give you an idea of the thoroughness and the way that Bibi and Peter approach this type of science. And so without further ado, I'm going to hand it over to Bibi and Peter.
And I decided to just introduce topic, why the study that we are going to present to you, why it was done. This is a meta-analysis of MS clinical trials that we have done several years ago. And it includes both relapsing, remitting, and progressive MS trials. And what this shows is that the efficacy of current drugs, FDA-approved drugs, on inhibiting disability progression decreases with advancing age of MS patients. This model, in fact, predicts that after approximately age of 53 years, our drugs do not do better than placebo in inhibiting disability progression. So if we just take conservative limit of 55 years, that leaves 45% of people currently living with MS without effective treatments. Additionally, I'm going to argue that these middle-aged people, aged 35 to 55, have relatively marginal efficacy on inhibiting disability progression. And I'm going to show it to you on this clinical trial of ocrelizumab in primary progressive MS that actually excluded everybody after age 55, recruited people with a mean age of 44, and reported 25% efficacy on disability progression. So the reason why I'm giving you ocrelizumab is that based on these age-adjusted residuals, it's one of the most effective, if not most effective, treatments we currently have. And here are the data from this clinical trial. And you can see that the maximum separation of the arms already happens at 12 weeks. And then the arms basically progress in parallel. So that tells me that the drug is working on biology that can be inhibited so fast, and that biology are contrast-enhancing lesions. And indeed, we can see that unusually large proportion of these primary progressive MS patients had contrast-enhancing lesions. So when we say that ocrelizumab had 25% efficacy, what does it mean? Does it mean that 25% of those treated patients will never progress? Or does it mean that people will progress on 25% less? Actually, it doesn't mean any of these things. It's a relative number, and it's a relative number to when the experiment was stopped, which in case of this clinical trial was stopped after 4 years. But you can get absolute number by selecting any level of disability threshold and simply asking, when do these two arms reach that threshold? And the difference in this time is 6 months. And it will remain 6 months forever, as long as these two arms are progressing in parallel. So in middle-aged MS patients with substantial MS lesional activity, ocrelizumab delays sustained disability progression by 6 months. Unfortunately, what doesn't remain stable are side effects, right? Infections and cancers, they become cumulative as we are giving these drugs to older and older patients. Not only that, because ocrelizumab actually inhibited contrast-enhancing lesions by close to 100%, and it does so always, it means that all of these people who progressed on ocrelizumab must be progressing due to different biology. And I'm going to call that biology non-lesional MS activity. And so I believe that if we want to treat these later stages of MS, we actually must develop treatments that target this non-lesional activity. And we would know that we are targeting non-lesional activity because these treatments should not only affect the intercept, but they should also lower slope, meaning the longer people are on these treatments, the more benefit they should sustain compared to the placebo arms. So I'm not going to talk about lesional activity because we really understand what causes that. It's a focal influx of immune cells from periphery into CNS tissue associated with the breakdown of blood-brain barrier. And this biology is really exceptionally well inhibited by current treatments. As I said, ocrelizumab does so by 95%, but many others do so phenomenally well. The reason why these drugs don't work is not because somehow they stop inhibiting lesions. It's because that biology by themselves decreases as people with MS progress. And as I indicated, therefore, what is driving this disability progression on people who are treated or on people in the later stages of MS must be non-lesional activity. Other people call it PIRA, progression independent of relapse activity. And of course, we, as a field, we study this using pathology. And pathologists obviously identified many different processes that happen in the CNS tissue when people die with MS. But it's very difficult for pathology to determine which of these are causal processes. And that is really the reason why we are studying cerebrospinal fluid biomarkers. Our hypotheses are that pathogenic mechanisms that underlie MS non-lesional activity are multiple. They are individually diverse. and they likely evolve with disease progression. And therefore, if we want to treat fully evolved progressive MS, it will require rational combination treatments that target ideally all disease mechanisms that are expressed in a specific patient. Both of these hypotheses come from our collective experience with all other complex polygenic diseases, such as, for example, cardiovascular diseases, where we today have hundreds of different drugs, and they are prescribed to specific patients based on longitudinal measurements of these processes that we used to call risk factors, but actually we now understand that they are truly causal processes and that they cause cardiovascular diseases through dose exposure interaction. And we gain this knowledge by our ability to measure these processes longitudinally in living people with cardiovascular diseases. And so similarly, I believe that we can only understand which of our candidate causal processes are truly causal if we learn to measure them in living people with MS. Furthermore, in comparison to cardiovascular diseases, we actually have to measure them in a way that assures CNS origin of biomarkers. Because as you can see, these processes are really happening or can happen in any organ. And with aging, they are likely to happen in all organs. And so I really don't know in any other way how we can measure these processes, but by measuring cerebrospinal fluid biomarkers. And of course, we glean pathology from these non-interventional studies that we are going to share with you today, but eventually we really need to also implement therapeutic trials and understand how drugs affect these CSF biomarkers and then truly validate these causal processes. So with that, I will let Peter take over and describe what we have done.
Okay, thank you, Bibi. So how did we go about identifying molecular mechanisms underlying MS disease? We did that first by measuring protein levels in cerebrospinal fluid. And this was achieved using a clever platform called SomaScan. This platform measures protein levels using DNA aptamers as opposed to using antibodies. These DNA aptamers are single-stranded molecules of DNA with modified side chains. They assume certain 3D conformation that allows them to bind with high specificity to their respective epitopes. And because there is very little cross-reactivity between different aptamers, they allow unprecedented level of multiplexing. So in our case, we were able to measure 5,000 analytes in very small volume of CSF. It was only 50 microliters. Now, at that time, this platform was not commercially available. It was proprietary to Novartis. That's why we came up with a cooperative research and development agreement between NIH, Novartis, and SomaLogic. And all this work was basically done under this agreement. So here is a design of our study. Patients with certain diagnosis of MS or suspected of MS diagnosis were enrolled into our natural history protocol, which includes collection of clinical, functional, and MRI imaging outcomes. Healthy people were part of this natural history protocol, and all these healthy volunteers underwent the same set of procedures as our MS patients. At the time of enrollment into the protocol, the cerebrospinal fluid was collected from MS patients and healthy people. The clinical CSF biomarkers were prospectively measured at NIH clinical laboratory, while the cell composition of the cerebrospinal fluid was assessed on 50-fold concentrated samples using multicolor flow cytometry. and then the cell-free supernatants were frozen for further use. The protein levels, as I mentioned, in these CSF samples, and we measured them in over 1,000 CSF samples, representing almost 400 MS patients and 45 healthy donors, were measured using this SomaScan assay. Additional biomarkers that have known abnormalities in MS were measured by in-house immunoassays. The group of healthy people that we enrolled in this study was important because it allowed us to regress out effects of natural aging and sexual dimorphism, as I will show you on the next slide. So all of these 5,000 somomers that we measured were adjusted for healthy donor age and sex effects, and then these adjusted somomers were used for the downstream analysis. So in addition, we also generated 9 machine learning models of different MS outcomes. Briefly, these models were generated by first splitting the MS cohort into training and validation. While the training was 2/3 big, the validation cohort was 1/3. And we were balancing for the MS subtypes in these 2 cohorts. The models were generated using 2 different algorithms, and their performance was tested in an independent validation cohort of samples. So because MS is a chronic disease that progresses with age, and there are differences between males and females when it comes to disease severity or progression, I would like to show you the importance of this age and sex adjustment that we did. And I'm going to show it on example of a protein called smoothulin. This protein is part of the SomaScan assay. And on this plot here, you can see correlation of smoothulin with disability outcome. In this case, we used outcome combi-wise. And you can see that the protein explains 11% of the variance of the disability in our MS cohort. But when you look at this plot, the same protein also correlates with age, explaining over 30% of variance. And at this point, we don't know if the accumulation of the protein is related to aging process or whether it's related to MS pathology. And when we regress out the age of MS patients, what you can see is that smoothulin levels adjusted for MS patients' age result in residuals that no longer correlate with disability. But because we have this healthy donor cohort, we can ask the question whether the protein correlates with age in healthy people. And indeed, when we look at that, the protein does correlate with age. And when we subtract this healthy aging effect from MS smoothulin levels, we will find out in this pink line that the residuals of these adjustments still correlate with disability with highly significant p-value explaining 6% of variance. Very similar approach was taken for adjustment for sex differences between males and females, and all these adjustments were applied to 5,000 somomers, and then these adjusted somomers were used for downstream analysis. So these days, it's relatively easy to collect enormous data sets, whether it's proteome, transcriptome, lipidome, or even whole genome sequence. But these enormous data sets that are information rich are really hard to dissect if you don't have some phenotype related to those numbers. That's why we needed good outcomes, good scales that can measure MS progression and MS severity in order to match them with the proteomic data from CSF so we can uncover the biology of MS. And that's why we spent past over 7 years working really hard on developing new sensitive outcomes that measure MS progression and MS severity. And with these granular outcomes, when we combine them, for example, with the protein biomarkers, we can ask the question, what is the biology of MS? So I'm gonna repeat a little bit what Bibi already showed. This is a simple scheme of MS evolution. At the beginning of the disease, new focal lesions start forming, and we can detect these as contrast-enhancing lesions on MRI images. They peak within some years of the disease onset, and then they go down either as a effect of therapy that we apply, or even without the therapy, they go down. The volume of total lesional volume in the brain can be measured by T2 lesion load volumetric outcome that basically quantifies the volume of lesions in the brain. This outcome starts accumulating at the disease onset and peaks around the time when the new focal lesion starts going down and remains relatively stable for the rest of patient's life. So the MS progression shows you the accumulation of disability over time, and it can be measured as cognitive disability or as physical disability. And as I said, we have developed multiple outcomes measuring progression of disability involving either brain MRI-derived outcomes such as brain atrophy, or different functional tests or clinician-derived neurological scales. I'm showing you on the right side 2 examples of these progression outcomes, CombiWise and brain damage. CombiWise focused more on physical disability progression, and brain damage more focused on cognitive disability. These colorful lines that you see are actually regression lines of multiple longitudinal visits of patients. So they are showing you the slope of progression of these patients, and you can appreciate on both of these images that as the age progresses, so does the disability accumulation. So because these scales are new and they are not really used by the outside world, we wanted to first make sure that they still correlate with disability outcomes widely accepted and used by MS field that are depicted in blue color here. So we correlated these traditional outcomes with our new scales in our cross-sectional MS cohort. And we can see that the T2 lesion load outcome and brain damage outcome correlate strongly with the 2 traditional outcomes measuring cognitive disability, while the spinal cord disability and CombiWise outcomes show strong correlation with 4 traditional outcomes measuring physical disability. So now that we have sensitive outcomes measuring disease progression and we have matching proteomic data on these patients, we can start asking the question, what is the biology that underlies progression of MS patients? And we can do this basically in 2 ways. The first way is biology agnostic approach that uses machine learning algorithms. As I will show you later in my talk or at the end of my talk, this approach is great for generating predictive models, but it's not so great in terms of teasing out biology for various reasons. The second approach takes advantage of all the accumulated knowledge of mankind about diseases and uses a tool, for example, like ingenuity pathway analysis. This tool contains over 8 million instances of manually curated knowledge about biology, genes, proteins, and their interactions that have been derived over time from human and animal studies. And this is the tool that we decided to use to tease out the biology out of our SomaScan data. So how did we do it? As I mentioned, we have 5,000 somomers that were adjusted for healthy donor age and sex. And these somomers were correlated with 4 disability outcomes: T2 lesion load, CombiWise, brain damage, and spinal cord disability. The correlation was done in the cross-sectional cohort of MS patients. And out of the correlation, we got Spearman correlation coefficient and corresponding p-value. Because we correlated so many somomers, we wanted to adjust for multiple comparison, and we did that using false discovery rate adjustment. And for somomers that passed FDR-adjusted p-value threshold, we took the Spearman correlation coefficients for these somomers, and we submitted these into IPA core analysis. So the ingenuity pathway analysis gives you then significant pathways, and they have associated z-score and p-value. This activation z-score basically tells you how well your data set matches the knowledge that is in ingenuity pathway analysis knowledge base. And the p-value tells you how confident was this z-score assigned to your data set. So when we look at the pathways identified for the 4 disability outcomes, and we summed up the z-scores that were identified by at least 2 progression outcomes, we found out that most of these pathways are activated, which you can see as red bars on this plot, and they can be divided into 2 major groups. The group number 1 is related to neuroinflammation and represents pathways like IL-17, IL-13, IL-6 signaling. But we also have signaling related to myeloid lineage like TREM1 or GM-CSF. We also see dendritic cell signaling and HMGB1 signaling related to inflammasome activation. The other set of pathways that we identified are related to fibrosis, and these are represented by pathways highlighted in green, overlapping pathways of hepatic fibrosis, wound healing, and pulmonary fibrosis, but also epithelial mesenchymal transition regulation by growth factors. We also identified pathways related to hypoxia, represented by HIF1 alpha signaling, that also can induce epithelial mesenchymal transition. So to sum up these findings, we see that MS progression shows increased inflammation. This finding goes against the current belief of MS field that was presented as lately as last year in Multiple Sclerosis Journal, but by International Progressive MS Alliance, that shows that MS inflammatory diseases in MS go down with MS progression, while we are showing here that inflammation goes up with progression. So because of this, we also wanted to make sure that what we see is real, and that's why we looked into a longitudinal cohort of untreated MS patients. This cohort was collected during our primary and secondary progressive MS trials that were placebo-controlled. So we had a nice set of samples collected in untreated stage. And what we did, we looked at SOMAmer levels between the first LP and the follow-up LP, and we calculated yearly change. So this would be the level how the SOMAmers are changing over time in untreated stage. And again, differences that passed FDR-adjusted p-value were submitted to IPA core analysis. And when we look at what we got, we were surprised, or not surprised, we basically confirmed that these pathways are very much similar to pathways identified in the MS cross-sectional cohort, even though in this case, in the longitudinal cohort, we were submitting to IPA yearly differences in specific biomarkers, while in this case, there were progression outcomes involved. So a question that still remains unanswered in MS field is why some patients have predominantly brain disease while other patients develop predominantly disease of the spinal cord. And I'm going to demonstrate this on an example of 2 of our patients. Patient 1 on the left is a 55-year-old male with almost healthy-looking brain, as you can see in this FLAIR image, with just 2 periventricular lesions depicted by yellow arrows here, while patient 2 is a 37-year-old male, and you can clearly see that there is ventricular and sulcal atrophy out of proportion of age for this patient, and there are confluent lesions in the white matter. But when we look at sagittal T1 images of these patients, we can see something different. The first patient with almost healthy-looking brain has a severe atrophy of the spinal cord, and the spinal cord is loaded with lesions. While on the other hand, the patient 2 with brain atrophy has very healthy-looking spinal cord, and there are no lesions in here. So because of our deeply phenotyped cohort of MS patients, we were in a great position to try to answer the question whether this is a stochastic process or whether there are some molecular processes that underlie these differences, why some patients have brain and some patients have spinal cord disease. So how can we do that? Well, we have outcomes that can measure cognitive and physical disability. In this plot, you see brain damage representing cognitive disability on x-axis and combi-wise representing physical disability on the y-axis. And these 2 outcomes correlate with each other, so we can draw a regression line among these points. But what you can also appreciate is that there are patients way above the line, and these would be patients that have proportionally higher physical disability. And there are patients below the regression line, and for these, we would say that they have more of cognitive disability compared to physical disability. And when we go, so when we hypothesize that when we take residuals of this correlation, we can isolate spinal cord disability outcome. And when we go back to the example from the previous slide, our two patients, you can see that patient 1 with normal brain and damaged spinal cord is located up here and has much higher physical disability, while patient 2 with atrophied brain and normal-looking spinal cord has predominantly cognitive disability and very little of the physical disability. So how can we tease out the biology of these differences? So we have this spinal cord disability residuals, and what we can do, we can isolate extreme patients in this group. And we can do it by simply selecting patients below the first quartile of the group distribution. These would be patients that have predominantly cognitive disability, and patients in orange above the third quartile that would have more of the physical disability. And we can use propensity score matching to pair patients from the blue group to patients with the orange group, making sure that they have comparable brain damage outcome, which you can see on this plot, but they have very different levels of physical disability, as you can see in here. But how do we know that the blue group and the orange group really have different levels of spinal cord damage? Well, we check that by looking at our MRI images that luckily extend caudally to C3 level, and we were able to use Spinal Cord Toolbox that contains an algorithm that can calculate an average spinal cord area between C1 and C2 level. So when we look at this fully quantitative outcome of spinal cord atrophy, that is depicted on x-axis, and compare it to our spinal cord disability outcome calculated as residuals of this relationship, you can see highly significant correlation between them, explaining 20% of variance. Moreover, when we look at our blue and orange group of patients, you can see that blue group that has more of the cognitive disability and should have lower spinal cord disability is characterized by higher level of spinal cord area, and on the other hand, the group with more spinal cord damage has a lower spinal cord area. So now that we have these two groups of patients, we can, similarly to what we did before, calculate differences in isomers between these patients, these two groups of patients, and submit the differences into IPA. What we found out was basically that patients with predominant spinal cord damage have highlighted activation of pathways related to fibrosis, represented here by FGF signaling, STAT3 pathway, pulmonary fibrosis, again, HIF1 alpha signaling. But we also see activation of pathways related to CNS response to injury, such as synaptogenesis signaling pathway here. On the other hand, patients that have predominant damage of the brain are characterized by neuroinflammation, specifically IL-17 related neuroinflammation, P38 MAP kinase signaling, but also the innate immunity as it is displayed here by crosstalk between dendritic cells and natural killer cells. And importantly, pathways that are activated in one phenotype are generally repressed or inhibited in the other phenotype. So now that we have identified processes that are associated with progression of MS, and also some processes related to differences between brain and spinal cord involvement, we were really interested in identifying biology that would be associated with MS severity. So when we go back to our little scheme of MS evolution, you see the purple line showing MS progression over time. And the MS severity basically is a measure of how bad disease patients have. So if we have two patients of the same age or of the same disease duration, and one of them would have low MS severity, it means that this patient accumulated low level of disability over that age, and patient with high severity would accumulate much more severity across that same time period. So how can we generate these MS severity outcomes? Well, we can do it by regressing age out of disability outcomes. An example is combi-wise severity outcome in this plot here. As you can see, combi-wise disability correlates nicely with age. And when we take residuals of this relationship, we will come up with combi-wise severity outcome. So how can we identify pathways that would be specific for MS severity? Well, first of all, as I showed you before, we isolated pathways that correlated or that were associated with MS progression by correlating SOMAmers with progression outcomes. We did the same for MS severity outcomes. So we took the 5,000 SOMAmers and correlated them with 4 MS severity outcomes. Now we look at these pathways identified in either one of these approaches, and we saw that there is an overlap between pathways, meaning some pathways are associated both with progression and with severity. But what we were interested in are really pathways that have stronger association with severity over progression. And so we did that, and we selected pathways with, because we have the Z-score, the activation Z-score, we selected pathways that have higher activation Z-score for severity, and therefore stronger association with MS severity, as you can see on the dots, red dots here that are below the one-to-one line, showing that the Z-score is higher for MS severity on x-axis, or for pathways that are inhibited, the selected pathways were above the one-to-one line, which means they are more inhibited with MS severity. So when we did all of this, we identified 25 pathways that have strong association with MS severity. The first group of them is related to adaptive immunity. More specifically, we identified strong B-cell signature represented by BAFF and APRIL-mediated signaling here. This signature was not identified for MS progression and seems to be specific for MS severity. But we also identified IL signaling pathways, but that signaling really showed stronger association with MS severity compared to progression. Interestingly, we also identified several pathways related to viral pathogenesis. We identified pathways related to innate immunity, such as phagosome formation, and again, crosstalk between dendritic cells and NK cells. We identified a new signature for pyroptosis that was not present for MS progression. The other novel pathway specific for MS severity was production of nitric oxide and reactive oxygen species in macrophages. Now, when you look down here, there are some blue bars which represent mostly pathways related to central nervous system, specifically synaptogenesis and neutrin signaling. And because of their negative Z-scores, it means that they are inhibited, and therefore we concluded that these pathways might have beneficial role for MS severity. Now, we were able to calculate IPA scores for these 25 pathways in our cross-sectional cohort of untreated MS patients. And using unsupervised clustering, we were able to generate 4 clusters in our cohort of patients. One of the clusters is the B-cell specific cluster, as I described before. Then we have additional 13 inflammation-related pathways down here. We have a cluster of beneficial pathways related to central nervous system, and a small cluster of containing activation of coagulation and beneficial pathways related to antioxidant properties of vitamin C. We also depicted clinical MS sub-diagnosis here, and as you can see, the position of the patient in one of these 4 clusters really has no association with what type of MS patients have. So lastly, I would like to briefly talk about the machine learning models that we developed in this project. This is a topic for a whole new seminar, so I'm just going to give you a gist of the story. As I mentioned at the beginning, the modeling was done in the training cohort, which represented 2/3 of MS patients. In this training cohort, we used 2 sets of predictors. It was either 5,000 SOMAmers or 12.9 million SOMAmer ratios. Both of these were adjusted for healthy donor age and sex. And we used these 2 sets of predictors to predict 9 outcomes in MS. contrast-enhancing lesions, 4 outcomes for MS progression, and 4 outcomes of MS severity. We use 2 basic types of algorithms here, a linear elastic net algorithm and decision tree-based algorithm called random forest. And each of these algorithms produce models that generated predicted outcome that was then correlated to the measured outcome. And out of this correlation, we recorded different metrics that would characterize how well the predicted outcomes correlate to the measured outcomes. And these metrics were collected for all training samples. And in case of random forest models, we also had these so-called out-of-bag samples. Then the model's performance was tested in the independent cohort of samples that in no way contributed to their development. And in this independent validation cohort, we collected the same set of metrics to evaluate how well the correlation between predicted and measured outcomes is. On the next slide, I'm showing you the overview of these results on an example of Spearman-Rho correlation coefficient between the predicted and measured outcome. It's a busy slide, but if there is one message I would like you to take from this, it is that the performance of this machine learning model in the training cohort is never predictive of how well the model will perform in the independent validation cohort. And I will demonstrate it on example of 2 outcomes, contrast-enhancing lesions and spinal cord disability. For contrast-enhancing lesions, you can see that the training cohort performance represented by these circles is relatively low. It was actually the lowest out of the 9 outcomes, while the spinal cord disability outcome has training cohort performance pretty high, close to 1, Spearman coefficient of 1. But when we look at the performance in the validation cohort, which is represented by these diamonds, you can see that for CEL outcome, the validation cohort performs almost as well as the training cohort model. On the other hand, when we look at the spinal cord disability outcome, the performance is much lower, significantly decreased from the performance of the training cohort. The other result out of this experiment was that the performance of the independent validation cohort in the model in the independent validation cohort is mostly dependent of how well your outcome measures the biology that you are trying to model. And out of these 9 outcomes, CombiWise is the outcome that in our hands has the highest sensitivity to measure biology. And as you can see, the validation cohort performance for CombiWise was the highest out of these 9 outcomes. So let me conclude all these findings that I presented to you. B-cell associated compartmentalized neuroinflammation increases with MS progression, and therefore there is a great need for development and testing of CNS penetrant anti-inflammatory drugs. MS patients with proportionally higher Th17 signaling develop predominantly brain damage, while fibrosis is associated with spinal cord damage. Neuroinflammation is the strongest determinant of MS severity, while processes like synaptogenesis and neutrin signaling preserve neurological functions and are beneficial. CSF biomarkers derived from one spinal tap can be assembled into different models that predict various clinical and imaging aspects of MS patients with accuracy that exceeds all published MS models. And our results support the need for biomarker-guided patient-specific polypharmacy regimens to limit non-lesional MS activity and MS progression. And at the end, I would like to acknowledge our group neuroimmunological diseases section, our lab members, and our great clinical team, all our patients and their caregivers, our internal collaborators from NIID Biostatistics branch that helped us with the machine learning part of the project, and of course, our external collaborators from Novartis and Somalogic, without whom this project would never be accomplished. So I thank you for your attention, and we are ready for your questions.
Mike, you're muted still.
You should stop
People
sharing.
can go and ask questions in the chat. I'm going to go ahead and ask the first question. I assume that when you see something like fibrosis and you're measuring proteins out of the CSF, that it doesn't necessarily have to be fibrosis. For example, TGF beta is a perfect molecule that you might pick up that would be labeled by a program as fibrosis. It would obviously have a lot of other functions.
Yeah, but the pathway is also, so the pathway is integrating the signaling molecule with molecules such as collagens. So turns out they are collagens in CSF, and they are increased in multiple sclerosis.
Oh, right. I mean, and certainly TGF beta, when you see gliosis.
Yeah.
Right. But
Yeah.
you might also see the same molecules as obviously regulating inflammation or Tregs or what, I mean.
Yeah, and you know, Peter showed you only pathways, right? The paper that is submitted also has the upstream regulators, and you know, there you can see that, for example, it's not only IL-17. There is clearly, you know, interferon gamma as an upstream regulator, but there is also, you know, IL-4, IL-13 as upstream regulators, right? So,
and do you feel like there's a question here about the clinical radiologic paradox, which, you know, could reflect gray matter involvement as a major source for cognitive impairment. How does that fit into this modeling?
So actually, you know, we didn't have a chance to show you everything, right? So this brain damage was actually PCA of SDMT and brain atrophy. So it has the cognitive information in it, right? And, you know, you see that there was a correlation, for example, between combi-wise and this brain damage. You know, correlation means, in general, for most people, these things tend to go together. But then, you know, these residuals that Peter was nicely showing you helped us to determine, right, what is the biology of predominantly, you know, cognitive brain damage versus what is the biology of predominantly spinal cord and physical.
Yeah, I mean, let me ask you, because this is one thing that always bothers me, is that if I was to say what I thought was the prototypical Th17 demyelinating illness, it would be NMO, and it's predominantly spinal cord. And yet your data suggests, and maybe it's just for progression, that IL-17 now is very important for progression in the brain.
Yeah, I think that I think that example that you gave, right, NMO, I think it also really depends what is the target of the immune response, not just what is the phenotype of the immune response, right? But I totally agree, but I'm sure if we did NMO, it wouldn't be just IL-17, it would be also neutrophils.
All right, I'm going to call on Ann Cross to ask a question. I think you'd have to unmute, Ann.
I keep unmuting and it keeps muting back by the host, it says, but can you hear me now?
Yes.
Okay, excellent. So I was wondering, I mean, one reason or thought I've had for many years, which may be totally off the wall, is that the myelin and other, you know, parts of the central nervous system too, such as astrocytes, are different between the spinal cord and the brain. Like the components have different ratios. And so I have wondered if that might explain some of the differences amongst people between predominantly spinal cord and predominantly brain. But I didn't see anything in your SomaScan results related to myelination processes. Did you pick up anything like that?
We did not, Anne, but to tell you the truth, I think it really depends what you are measuring, right? So, you know, there really, there are few true myelin proteins or oligodendrocyte proteins in SomaScan, such as, for example, opalin, right? If you go into Human Protein Atlas, opalin is pure oligodendroglial protein. And it's measurable because it has a good signal to noise ratio. It makes absolutely no difference between MS patients and healthy donors. So clearly, you know, it depends what is the function of the protein, whether it's released, right? And I would say at 5K version of SomaScan, I don't think you have proteins that would answer your question. So I don't think that not having it in our results means that that hypothesis should not be explored further. You know, from 5K to 7K, I can clearly see many more CNS proteins. So the biggest increase in the assay are really CNS proteins. So I think that 7K may be quite interesting.
Okay, well, anyway, this is fantastic and really getting at the cause and of the disease and its progression, which is great.
And, you know, I would say back to your comment, I don't think we know what is the egg and what is the chicken, right? So maybe if you have injury in the spinal cord, maybe the biology is such that you're going to be driving fibrotic signature, right? And that would totally go back to that idea that, you know, the proteins and the biology is different in the spinal cord than it is in the brain.
But you know, that's interesting. Have you looked at disorders like MOGAD and NMO that reported supposedly don't have progression between attacks? Because to me, that's obviously a big difference between MS, and says that there is something fundamentally different about what's going on and how MS brain and spinal cord handle injury.
So, Mike, it's really difficult for us to get longitudinal CSF from non-MS patients. The reason why we get longitudinal CSF on MS is that we also run clinical trials where longitudinal CSF is part of those trials. You know, we have an option for patients to give us CSF, and there are some patients that even in natural history, and, you know, for example, I don't know if it came through, but we have longitudinal healthy volunteer CSFs, you know, in this data set. But we just don't have longitudinal, you know, NMO CSFs. So I would love to answer that. And yeah, it's a terrific question.
Is there a way to, you know, when you say that you're trying to be closer to the CNS by using CSF, what about the people that are studying, for example, astrocyte responses by looking at exosomes in the blood?
Well, Mike, we are.
I mean, at some point, you know, I mean,
yeah, Mike, we are those people too.
Yeah, I know.
Okay, and you know, we haven't published. We're still working on publishing, but what I can tell you is we have used glass, we have pulled out so-called astrocyte exosomes, but we did experiment that nobody else did, and that is we ran the proteomic analysis on those so-called astrocytic exosomes, and we pulled out granulocytes. So then we went back to Human Protein Atlas and found out that glass is astrocytic protein in the CSF where you don't have anything else, but it's actually present in, you know, azurophilic granules of the granulocytes if you are taking it from blood. So, you know, NFL is obviously fantastic. If you have purely CNS protein, right? I still believe, we will enhance proteomics from blood, but we will not be able, I think, anytime soon determine, whether oligodendrocytes are dying out of apoptosis from the blood, right? Because, apoptosis signature is not oligodendrocyte specific. So unless the vesicles really pan out, and clearly there are many wonderful groups working on this, us included. You haven't seen anything published from us, not because we haven't been studying it in detail, but because we are seeing that it's not as simple as people are trying to make it.
Very good. Are there any other questions from the audience? Bob, do you have any closing remarks? You're on mute.
See, I actually have a question first. I'm just wondering, Bibi and Peter, do you think that a CSF assay should become a standard part of a workup for MS going forward?
Absolutely.
My answer is, we are subjecting ourselves to colonoscopies, which is invasive procedure, and we agree to put ourselves under anesthesia, right? Because it has a proven clinical value. To me, this thing has greater value than my gut, even though I'm quite hungry now. So I think if we can show that CSF biomarkers have proven value, not just for MS, but for all CNS diseases, why not? I think we should do that. And, Peter and I are healthy volunteer 1 and 2 out of this healthy volunteer cohort. In fact, what you saw on that picture was me doing LP on Peter. And I get LP Thursday morning, I get NIH breakfast, and I go and see patients Thursday afternoon, right? It's not a big deal. If it is done correctly, it's not painful, it's not a big deal procedure. It's really, we made it into this big deal procedure, and I believe we made it into that because it's not reimbursed. You can't make any money out of CSF procedure. That's the reason, nothing else.
Well, thank you both for a terrific presentation, and thanks to the audience and to Mike, and particular thanks to Banner Life Sciences for sponsoring the MS Research Exchange and today's event. We really, you will get a survey when you sign off. We really do need your feedback on this experience, so we'd really appreciate that. You could quickly fill that out. We're really hoping this is only the beginning of the discussion about this remarkable work that Bibi, Peter, and their team at NIH are doing. The slides and the paper are online for any questions or discussion or comments you want to make after the event. We really hope that MS Research Exchange can provide a place where that conversation continues. So thanks very much, and appreciate your time.
Thank you. Bye-bye.
Bye.